Do you remember the experience that set you on the path you're on today? For me, there were two such experiences. The first came in my organic chemistry class in undergrad. We were tasked with designing and synthesizing a molecule that we predicted would have antibacterial properties. We didn't succeed in creating the next great antibiotic because we didn't really know what we were doing. But we were introduced to a really important idea, that the basic science we were learning and doing every day could have implications in solving a major health problem. In the last 30 years or so, the number of new antibiotics that have been approved for the treatment of bacterial infections has significantly decreased. In the same time period, the number of bacteria exhibiting antibiotic resistance has exponentially increased. This means that we're running out of antibiotics to effectively treat all types of bacterial infections. So we need to dedicate time, money, and resources to either discovering or designing new antibiotics. The second experience that led me to my current research was discovering that I was passionate and interested in RNA biology. We often think of RNA as this intermediate between DNA and protein in the central dogma of molecular biology. But in fact, most RNAs in the cell aren't directly involved in this process. RNAs have the ability to fold into unique structures, which allows them to carry out many diverse functions in the cell. Some RNAs are able to have catalytic properties just like proteins. And other RNAs, most interestingly to me, can act as regulatory elements. The RNA regulatory elements that I study are called riboswitches. Riboswitches are RNA regulatory elements that modulate gene expression in response to changes in the concentration of a cellular metabolite. And usually, the protein product of these regulated genes uses that small metabolite as the substrate of their reaction, or it is the product of their reaction. Riboswitches are primarily found in prokaryotes, which makes them a potential target for antibiotic development, and actually a pretty good target. One of the problems we have with current antibiotics is that they have off-target effects, which lead to adverse side effects in humans. But if we were able to design a molecule to just target this riboswitch system in the bacteria, the likelihood of off-target effects and side effects would be reduced. The riboswitch that I study is known as the T-box riboswitch. And it's unique in that it responds to the whole tRNA molecule instead of a small metabolite. And more specifically, it responds to the charging state or the presence or absence of an attached amino acid. And the genes that are regulated by this riboswitch are the genes involved in catalyzing the reaction that adds the amino acid to the tRNA. And these genes are essential for cellular function. So if we disrupted one of these genes, we would kill the cell. In the absence of an attached amino acid, an anti-sequestrator helix is formed. This exposes the Schindel-Garno sequence, which is the site in bacteria the ribosome binds to initiate translation. Conversely, when there is an attached amino acid, a sequestrator helix forms. In this helix, the Schindel-Garno sequence interacts with an anti-Schindel-Garno sequence, which hides it from the ribosome, and protein synthesis doesn't occur. And this is the off state of the riboswitch. In a healthy, happy cell, most tRNAs contain that attached amino acid, making this off state the default state. And this makes the T-box riboswitch a particularly good target for antibiotic development because it's easier to keep something off than it is to turn something off. It's easier to stay with the status quo. Additionally, in bacteria that use the T-box riboswitch to regulate their essential amino acid-related genes, there are several copies that are unique of the T-box riboswitch. So the likelihood of all of them simultaneously developing mutations that lead to resistance is unlikely. So even if one of them mutates, you still have other copies that are being targeted, and your antibiotic is still effective. In order to design an antibiotic to target this riboswitch, we need to understand how all of the features work together to recognize tRNA and result in the switching mechanism. When I entered the project, we knew about 3 interactions between the riboswitch RNA and the tRNA. The first occurs between the 3-nucleotide anticodon of the tRNA and a complementary 3-nucleotide sequence found in stem 1. And this interaction is the interaction that's involved in choosing the correct tRNA for the regulatory mechanism. And the correct tRNA is the tRNA that has the same amino acid identity as the gene that's being regulated. The second interaction is an interaction between the top region of stem 1 and a region of the tRNA known as the Trna elbow. This interaction is also involved in picking the right tRNA for the mechanism and also plays a role in forming the right global structure of the riboswitch to facilitate the regulatory decision. The third interaction is the interaction that can only occur in the absence of an attached amino acid. And it's this interaction that stabilizes the anti-sequestrator helix and exposes the Schindel-Garneau sequence. We have some evidence that designing a molecule to compete with this interaction and force the riboswitch into the off state could be a good path to designing an antibiotic that targeted this riboswitch. The stem 2 and 2AB elements, which are found between stem 1 and this anti-sequestrator or sequestrator element, are important for the function of the riboswitch in the cell. When we introduce mutations that disrupt these elements, we kill the riboswitch. However, we didn't know what the role of these elements were in the regulatory mechanism. We didn't know if they were involved in directly contacting and selecting the tRNA, or if they just played a role in that global architecture to make sure that the regulatory decision happens. Through my project, we were able to determine that both stem 2 and stem 2AB contribute to tRNA affinity. And using a series of biochemical assays that allow us to detect direct contacts between RNA molecules, we were able to identify 2 new interactions. Shown here, I have a model of the riboswitch RNA and a model of the tRNA to indicate the new interactions. Shown in green is that previously identified interaction between the anticodon of the tRNA and stem 1. The nucleotides highlighted in red and indicated with the arrow are the first new interaction. This interaction occurs between stem 2AB, which folds into a structure known as a pseudoknot, and a region of the tRNA known as the D loop. In blue, I've shown the other interaction. And this interaction is between a motif in stem 2 that is known as an S-turn motif and a region of the tRNA known as the T-arm. In the literature, there are no examples of pseudoknot elements or S-turn motifs being involved in tRNA recognition. So through this work, we have shown a novel mechanism of tRNA recognition, as well as provided the first biochemical evidence for the role of the pseudoknot and stem 2 in the riboswitch mechanism. Furthermore, in the literature of structured RNAs, we do not see pseudoknots or S-turns facilitating RNA-RNA interactions. They often act as protein binding sites for RNA-protein complexes. These facts make these elements particularly good targets for new antibiotic development in the T-box riboswitch. One, because they're not found in other RNAs, so the amount of off-target effects is likely to be reduced. Additionally, we know that since disrupting these elements via mutation kills the riboswitch, if we're able to disrupt these interactions in the same way using a small molecule, we would think we would also kill the riboswitch. In order to effectively design an antibiotic to target the riboswitch, though, we first need to understand these interactions at the molecular level. We need to know exactly what's interacting with what. Additionally, we need to know if these interactions are specific to this one T-box riboswitch that I have been studying, or if they're found in all of the different T-box riboswitches which recognize slightly different tRNA molecules. So moving forward, in order to do this, I'm working on solving a three-dimensional atomic resolution structure of the complex to further define these interactions and identify their potential to be antibiotic targets. I hope that through my research, I'm able to help combat this antibiotic resistance problem a little bit. But there are things we can all do every day to help with this problem. The first is to just stop using antibacterial hand soaps. The CDC recently published a study where they showed that those antibacterial hand soaps were no more effective than just generic regular hand soaps. They even went so far as to ban some of the ingredients, those active ingredients that made them antibacterial hand soaps. So just stop flushing those antibiotics down the drain unnecessarily. The second thing we can do is to try to buy meat products that are labeled as antibiotic-free. Animals are often fed antibiotics as a part of their diet to keep them healthy, rather than only using antibiotics to treat them when they become sick. These antibiotics lead to increased bacterial resistance in the organisms, as well as spreading that resistance into the environment. So I hope that you take from this that we can all do a part in combating this antibiotic resistance problem by using antibiotics responsibly and being aware of the science, even the basic science related to this field. Thank you.